Ebola virus matrix protein VP40 and its role in viral budding. Ebola virus is a highly infectious RNA virus which causes severe disease in humans. Its symptoms are characterized by high fever and internal bleeding. It is composed of a viral envelope, a single-stranded RNA genome, as well as various viral proteins. Recently, Ebola virus has become an increasingly large public health concern. The 2015 outbreaks in West Africa killed thousands of people and devastated local health communities. Not only is Ebola virus highly virulent and transmissible, the response to Ebola virus has suffered from a lack of effective vaccines and antiviral treatments. Not a single vaccine or antiviral drug has been approved yet for Ebola virus. To better understand the virus and possibly develop new treatments, scientists have turned their attention towards Ebola matrix protein, known as VP40. Ebola matrix protein, found on the inner membrane of the virus, coordinates exit of the virus at the host cell membrane. While invading the host cell and replicating its genetic material, Ebola virus expresses matrix protein as well as other viral proteins using the host cell machinery. Matrix protein then localizes at the host cell membrane, where it allows for newly assembled virus particles to bud from the host cell and escape to infect other cells. Since VP40 plays such an essential role in the Ebola life cycle, many studies have been done to better elucidate VP40 structure and how it coordinates viral budding. First, in order to migrate to the host plasma membrane, two VP40 monomers must form a VP40 dimer. The dimer is shown here, with the two monomers forming a butterfly shape. Dimerization occurs as a leucine residue on one subunit, labeled in yellow, extends into a hydrophobic pocket formed by histidine, alanine, methionine, and phenylalanine residues. The interaction of these hydrophobic residues locks the dimer into place. As the dimers begin to arrive at the cell membrane, a patch of basic lysine residues found on the C-terminal domain of each subunit play a crucial role in interacting with the negatively charged inner cell membrane. Once VP40 dimers have migrated to the cell membrane, they begin to assemble even further into hexamers, each composed of 3 dimers. These hexamers are connected through hydrophobic interactions at their C-terminal domains. These hexamers then form long continuous filaments that line the host cell membrane. How does VP40 polymerization at the membrane actually help induce viral exit? Studies show that a hydrophobic loop on the VP40 C-terminal domain, labeled in magenta, penetrates 8.1 angstroms into the plasma membrane. This deep penetration into the host membrane, along with tight packing of VP40 filaments, makes it energetically favorable for the membrane to bed into a sphere. This makes it easier for subsequent viral budding and exit to occur. So based on the structural information we know about VP40, does it have potential as an antiviral drug target? Experiments show that mutating just 1 or 2 of the key amino acids that are responsible for VP40 structure or membrane interactions can severely reduce viral budding from host cells. For example, in the experiment shown here, key hydrophobic residues on VP40 that are involved in host cell membrane penetration are replaced with other amino acids. When the unmutated or wild-type VP40 protein is expressed in cells, large quantities of virus-like particles, labeled with green fluorescent protein, bud and exit the cell. In contrast, when the mutated protein is expressed in cells, localization and viral exit at the host membrane are severely hindered. This is quantified here, where mutants with amino acid substitution in the membrane insertion domain produce much fewer virus-like particles compared to the wild type. Experiments that have mutated other key parts of the protein, such as the VP40 dimeric interface or hexameric interface, produce similar experimental results. Additionally, VP40 controls other functions in the viral cycle that may be attractive drug targets. For example, VP40 disassembles into an octameric ring composed of 8 monomers, which binds RNA in order to regulate viral transcription, although the mechanism by which this occurs will still need to be deduced through future studies. Furthermore, the exact mechanisms by which VP40 induces membrane budding are still unclear, but evidence strongly suggests that certain domains on VP40 recruit the host cell machinery for membrane budding. This means that making drugs that target the host cell machinery required for viral exit is another possibility. Ultimately, with a large number of different protein interfaces and key amino acids that matrix protein requires in order to assemble its functional states, It is likely that inhibition of viral budding from host cells may become a viable answer to Ebola outbreaks in the future.